Katie, our member, is also formerly butterfly conservation, but Katie says she's worked in the nature conservation sector since 2006 with a focus on biodiversity monitoring indicators, trends, and also consultancy work on recreational disturbance with government agencies, local authorities, and NGOs. Prior to joining NatureMetrics, Katie was senior data ecologist with butterfly conservation, working on data strategy to improve the gathering, curation, dissemination of lepidoptera data for greater impact. Right, so very simple title to this talk. Katie is going to tell us about giving nature a number. So thank you very much.
Thank you. Can everyone hear me okay? Hopefully. Well, thanks, Rachel. You made me a bit emotional there. As I said in the comments, it was a team effort, but it's amazing to see how far that project has come and can't wait to see more in the future. But I'm here today talking in a different capacity, which is about DNA-based monitoring. And so Ian couldn't make it, but I'm really pleased because I wanted to do this anyway. So yeah, I've spent a career very close to data, as you can tell from the last talk. And my last job was working on the data review at BC. I've always been very interested in the future of biodiversity data, and last February curiosity got the better of me and I joined NatureMetrics. So I've since been working on projects that relate to looking for fish or fungi or newts or nematodes, all sorts of things. It's been a crazy ride, but hopefully I've got some interesting case studies and stories for you. So just quickly about NatureMetrics. I joined, as I said, last February, and there are 37 staff. We're now over 120 employees, working with over 450 clients around the world in more than 80 countries. And I know that as of yesterday, this map is out of date because we've just started working in Kazakhstan. But let's go somewhere more meaningful or understandable to most of us in the UK, and that's my focus. I work with UK conservation clients and rewilding particularly. So we're working with, I think, 14 wildlife trusts, lots of interest in freshwater detection of species and soil. The Rivers Trust, we've got 17 of them at the moment. But we've also got some really interesting projects with lots of people you'll recognise. A really great one with the Scottish Government trying to develop novel approaches to biodiversity indicators. National Trust seeing if we can detect dung beetles from, yes, dung. And the Freshwater Habitats Trust, a big citizen science project looking at 70 water bodies across the Brex. I didn't even think there were 70 water bodies on the Brex, but anyway. So what makes biological recording tricky? Well, and where do we fit into the picture and the future indeed? So we tend to think of biodiversity as being made up of two different, very non-scientific groups, both of which have measurement and monitoring challenges. Firstly, there's wildlife, which could be hard to observe. So there might be specific monitoring requirements. Time of day springs to mind, moths versus butterflies. Equipment, moths. You can tell where my experience lies. Elusive behaviour, moths. Weather conditions, butterflies, moths. And skills to detect the species, so everyone who's interested in Lepidoptera. And then there's the small things, which are potentially hard to identify. So think of soil fauna, fungi and bacteria, and maybe some of those less charismatic invertebrates, flies and things like that. So both of these types of biodiversity suffer from a lack of sufficient data for good decision making. And even in the UK, so one of the most intensively recorded collections of islands in the world, I imagine, maybe apart from the Galapagos. And I know that I come from the Isle of Purbeck, so we have a renowned biodiversity hotspot here. But even here, we still have a recording problem for the groups that aren't targeted by conventional recording. So DNA metabar coding can help with some of these problems. So giving data at large scales across the tree of life. It doesn't solve all of the biodiversity measurement problems, but it can do a lot for species detection and discovering the small things. And that's what I'm here to talk about today, is using Metabar Coding to detect wildlife, but also the small things that are difficult to identify, but that really run the planet, I think. So the interesting part, what is it that we actually do? So we've got a huge lab in Guildford where all of this happens. So we can basically ask and answer two broad questions from sampling from DNA. Firstly, we can ask, is species X present? So many of you will be familiar with the DNA-based testing for great-crested newts and also COVID-19. So that method's called quantitative polymerase chain reaction, qPCR, and that targets one species. So we can use this for a variety of invasive species. But more interestingly is, well, from an ecological perspective, is Metabar coding. And this is what I call community analysis. And this is asking which species are present. And we can apply a variety of different analyses, tests, assays, are all different ways of describing them, to DNA collected from water or soils and sediments or bulk catches of insects. And yes, this can include moths, to answer the previous question. And in-field sequencing isn't actually that far away, but not something we can offer today. So DNA metabar coding lets us take complex environmental samples. We can sequence all the data that comes from a particular taxonomic group using an assay, and we can set the net, the assay specificity, wide or narrow, depending on our aims and priorities. So, for example, we might want to look at all vertebrates in a sample, and I'll be showing you some examples using vertebrate assays, or we might just want to focus in on fish. So the next step is to match the sequences against reference databases and to identify them, and this is where you actually get the list of species from each sample. Just briefly, so within each of the three types of sample that we analyse, so water, soil and bulk invertebrates, we actually capture and detect species from two types of DNA. And I always forget to say this. So organismal DNA is the whole or partial bodies of organisms in the sample, whereas environmental DNA or eDNA are the traces of organisms that are not themselves in the sample. So this might be skin, feces or mucus. This is where the E in eDNA comes from. So how does it work? Well, this is our approach. At NatureMetrics, we help design the sampling, provide the kits, training, and for project staff or contractors, volunteers. And for water sampling, this is what it looks like. You connect the filter, which I've got one here, to a massive syringe. You filter water through it, you apply a preservative solution, and you send it back to us. Then it's about 8 weeks until you get your list of species. But just before we move on to the case studies and look at what some of the data looks like, I know as you are a very learned group, you will want to know about the caveats and questions, and I've tried to summarize them here. The considerations to have when thinking about eDNA are abundance, the issue with abundance, detectability, how effective the assays are, and bioinformatics. So firstly, we can't provide abundance data. In the future, we will be able to provide individual species abundance changes over time, but due to differences in detectability between different species, it's hard to say whether one species in a sample is more abundant than another in a sample, because it could just mean that they are releasing more DNA into the sample than another species or another individual indeed. So on detectability at this stage, the actual sampling level, it depends how sluffy a species is. This is the word I've made up. So that, to me, that's how much they shed their DNA into water, how long ago it shed, and in flowing water situations, how fast the water flows that the animal shed into. So life stages are not generally discernible, but spawning events for fish are, which is quite exciting. So we get a lot of people wanting to know about spawning events. So then there's this stage where the DNA is extracted and then the assay is used to amplify the target group, the DNA from the target groups. So at this stage, there's a bias in the effectiveness of our assays. So for example, we've got a fish, we've got a vertebrate assay, we've got a fish assay, but elasmobranchs aren't picked up that well from our vertebrates assay. So we're going to design a more targeted test for sharks and rays. Then last but not least, there's the bioinformatics. So this is the bit where the DNA comes out and we use an external database to look up and return the species. And so you'll be hearing more from Liam, hopefully. He's not the person who can't make it, and the Darwin Tree of Life project after me. So we depend on these databases to be able to return a species list from a sample. So whilst the coverage of the species is good in the UK and set to improve by projects like the Tree of Life, it's not the case for the whole world. And hence we are developing some taxonomy-free approaches to characterising communities and tracking change, which I'll talk about later. So just a few examples on wildlife detection, and I will go straight in. The first one is actually a case study from Sweden, and it's a coastal fish survey where the client, who's called Aquabiota, wanted to test the effectiveness of an eDNA approach to detecting fish species compared to conventional overnight netting. So they compared their netting to 1 3-litre sample of water pushed through one of these filters. And in every case, the eDNA sample detected more of the overall fish community than the netting did. The numbers on here represent the number of species detected at the sampling locations. So this kind of comparison is shown over and over again, both in our work, and we've got lots of other case studies like this, but also in the scientific literature. But it's not just fish species that live in the water whose DNA is found there. So what we're finding is analysing DNA from water samples can actually give better information on also terrestrial vertebrates. And there's a lot of evidence coming out about this form of data collection being more effective than camera traps. I like the title of this article, Fishing for Mammals. So back to the UK. So this is a National Trust case study that shows what happens if we cast the net a bit wider, and instead of focusing on just fish, we look at not invertebrates, we do invertebrates, but vertebrates. So these results are from a project led by the National Trust. So they've been sampling at ornamental lakes on the grounds of stately homes in England, and you're not going to be able to see the data, but what it shows is that the scale of diversity that we could recover just even here in the UK. So in total, across 40 samples, we got high confidence of records for 74 vertebrate species. So we got loads of fish, but we also got 4 species of amphibian, common frog, common toad, 2 species of newts, we've got 33 species of bird, and 22 species of mammal, including otter, water vole, badger, fox, deer, mice, and even a mole. So water courses are great DNA traps, and mostly because they're toilets, actually. So even from water, it's possible to profile much more than just the fish. And this is the first step of thinking more broadly with eDNA. So yes, we could use it to detect key taxa, but it can actually start to look at whole communities. There we go. So just another one that's a bit further afield. This is an old case study, but one of our favourites. It's from the Peruvian Amazon, and it shows the power of eDNA at the landscape or riverscape level. So in 2018, WWF Peru did a river dolphin survey along about 1,000 kilometres of waterway in the North Peruvian Amazon. So this was one sampling trip that took 160 samples. And not only were they able to map the presence of the river dolphins and also manatees, their main focus, but so from that one vertebrate analysis, they captured data on 650 species, jaguar, tapir, and even bats. And to get this kind of data would have cost millions of pounds, really, and taken a long time, but it took a few weeks, cost a fraction. And you know, it's hard to think about there being unsurveyed places in the UK, but you can see when in foreign out in the rest of the world where there's less intensive recording, it can be really powerful. So my analogy for this kind of project is that you go to the shop on an empty stomach and you want a loaf of bread, but you come home with a banquet for 10. I just have to quickly mention, whilst on water, the Ebio Atlas project. So we've discussed how, the UK is relatively well recorded, but there are still gaps. Well, as I said, those gaps are even greater across the world, and that's why we're looking to scale up DNA-based monitoring across the world through this programme. And it's a collaboration between NatureMetrics and the IUCN, and we're creating a global scaffold of freshwater eDNA-derived data that will map the world's biodiversity and lay the foundations for the global biodiversity framework. It's a large ambition, but we are actually getting there. And one of the most exciting things is that we are raising money to collect these global samples, but also encouraging those people who are doing their own monitoring to contribute their data into the Ebio Atlas so that it can be shared to GBIF and the Red List and made available for research and conservation. So it's good news all around. So moving on from improving wildlife detection to the small things that run the world and characterizing communities, quantifying success over time. And I think this is where I think DNA is really exciting, because it's the only way to get this kind of data. So firstly, an example from using flying insects to think about communities. And this is a forest research project. We had 15 forest sites, some with pure oak, some with pine and some mixed. And we collected several samples of insects in each site and plotted the data in an ordination. It showed us a couple of interesting things. And one is that insect communities in the mixed crop sites were just the same as communities in the pure oak sites. So it means the pine specialist species are not moving into these sites. The second is that the two pine sites that you can see are quite different from each other. So pine in the blue, one there and one there. Hopefully you can see my pointer. They have species that don't occur elsewhere. So actually, although these sites might not have the highest number of species, if you lose those ones, you could lose overall biodiversity from the landscape. And this kind of data collection is enabling us to sort of build thinking about how to appraise at the community level. And Michael rightly said that the future is this kind of targeted, repeat capturing and characterizing of communities from my perspective, not just detection of certain groups. So I'm really keen on this. And then going further down, one step down the size chain, we have a huge growth in interest in soils, and I'm really rather passionate about soils, and this especially coming from rewilding projects, because you could imagine a rewilding project starting with an intensive improved field, and they've got all these grand plans. They're very interested to know how the soil biodiversity is going to change. So we've actually worked out that we can detect a surprising amount of wildlife from soil. So this is the picture you can see here, is vertebrate eDNA from soil and sediments. It's all rather new and exciting, but I said I would actually talk about the small things. So in soils, we mostly focus on the organismal DNA of the small things that make up soil communities themselves, because these can tell us a huge amount about how ecosystems are changing. So we're looking at things like soil fauna, mites, springtails, insects, and loads of tiny worms that would be very difficult to analyze any other way. We're also looking at fungi, bacteria, and they're so diverse, tiny, and poorly known that they just can't be monitored in any other way as well. So Metabar Coding can unlock these communities for the first time and use them to monitor ecosystem change. So when we do analyses of soils, we can generate massive lists in the thousands of bacterial and fungal species, or what we call operational taxonomic units, but that doesn't really mean much to anyone. So what we're trying to do is look at how the overall communities compare, like we were with the forest insects, between different habitats. And we can use plots like this to show how similar the communities are to one another and whether this is changing over time. So it's really useful for tracking the success of restoration practices or intervention. And I think this is really the power behind DNA-based monitoring, because we're able to characterize whole communities, plot them on a scale from degraded to the target healthy habitat. This is where we're getting to. This is the golden ambition, is being able to place a site or a sample on this gradient. And we're getting there. We've actually just got a product called Restoration Tracker, which is based on a machine learning that teaches a model what is good and what is bad in terms of a condition. And we're starting to play with this kind of solution now. But it's not just soils that we can apply this approach to. A whole community approach is attracting lots of interest in coastal marine habitats. So for kelp, mangrove, seagrass, coral habitats, it's likely to be the bacterial communities that provide the data and power to track ecosystem health and determine signature of good. That's all rather exciting and a long way from actual biological recording, that last bit, I'm afraid. But if you want to speak to us more about any kind of species detection, then this is the team, and we're here to talk about anything, potential projects, about what eDNA can do and what it can't do. And yeah, thanks very much for listening.
Thank you very much, Katie. I think there were some questions on the chat. I don't know whether you can see them.
Yeah, I can. Oh, no, it is Liam. Damn. Do the results. So the first one is about data flow.
Not coming through at the moment.
Can you hear me?
Yes.
So the first question is that from Kieran about data flow? I have it on good authority that there are.
Just for the sake of the people in the audience.
Oh yeah.
Oxford, you might need to read out the question.
Thank you. Right. Do the results on NatureMetrics and other eDNA analyses get included into recording data sets such as local environmental record centres, national recording scheme societies? So it is, from our perspective, apart from the bioatlas, which I explained, that data that goes through the bioatlas will go to the NBN and to GBIF. The people that we work with, it is up to them to put the data in. We don't take responsibility unless they've said we'd like our data to go into that flow. But I think the ideal would be that they all did go into. The one thing that's interesting is eDNA-based derived data is slightly different, and there is a standard for it. But I think there's 12 records in the NBN from eDNA, so it's still a bit of work to be done. And next one, can you use eDNA? So this is from Graham Wally. Can you see eDNA methods being used to identify local sewage disposals in watercourses by local citizens? And have you been attending our weekly meetings? We have got some prototype solutions to being able to identify the source of microbes in the water. And we're working with a few water companies to develop this further. So yes, watch this space. There is a bit of a scary area, being the people who have discovered sewage outfalls though. So okay. Paul Seligman, when might it be practical and affordable for non-professionals to sample their garden pond or compost heap and know what's in it? If done periodically, could it indicate the impact of improvement measures? Yes, so we do have citizen science kits, and citizen scientists do measurements of their ponds and things like that. If you have a specific request, then do get in touch. I think over time the cost of the analysis will come down, but having been in the labs and seeing the infrastructure that is required to generate the data, I do understand why it's priced where it is priced. So should I keep going, or?
I think it's time for one more question, and then there's one question in the hall here. Martin whizzing down there with the microphone.
Hi. I'm fascinated by the soil side of your work there. With your barcoding, I mean, you've obviously identified a number of organisms you can track for in soil. Now, what percentage of soil, say bacteria, are you actually picking up in your barcoding? Because many of the bacteria in the soil are quite difficult to extract anyway, and sort of taxonomically identify. It's always been one of the challenges. Is there?
I can't answer on bacteria. Sorry about that. I have to follow up. But I can on fungi. In the UK, we can get 30 to 40% of soil fungi to genus level. Is that right? I think.
Right, thank you very much. Thank you, Katie, for a mind-blowing presentation.
Oh, great. Thank you.
So thanks to Katie. Now we have got our last speaker for the afternoon,